Hi, I'm Yidan, a PhD candidate from the Department of Earth and Environmental Sciences. Today, I will present my study on how multi-omics gut microbiome communities link metal exposures to children's neurodevelopment in the Canadian birth cohort. The pediatric gut microbiome plays a very important role in neurodevelopment through the gut-brain axis, and it is influenced by environmental exposure like metals, making it a potential interface linking metal exposure to adverse neurodevelopmental outcomes. However, most studies only focus on individual microbes, even though they function within interconnected ecological communities. We therefore hypothesize that the gut microbiome is organized into tightly connected species communities and functional modules, and these ecological units are associated with metal exposures and neurodevelopment in children. So we use data from the gestation and the environmental birth cohort in Canada, including children aged 8 to 12 years old. Stool samples were analysis used metagenome and metatranscriptome sequencing to profile gut microbiome species and functions, along with 19 metal elements being detected in the stool. Neurodevelopment was assessed using behavior scales and brain functional MRI, and social demographic and lifestyle factors were adjusted for covariance. To identify microbiome communities, we developed a low-end community detections with a threshold updating framework. So first, we construct correlation networks among species or functional pathways, applying threshold for correlation coefficient from 0.4 to 0.9, and detected communities at each level of the network. Then, when feature was overlapped between communities, the communities from the lower threshold will be replaced by those from higher threshold, yielding a final set of non-overlapping communities. Each communities and functional modules were summarized use the first principal component. And then we will do downstream analysis to assess their associations with metal exposure and neural health. From this framework, we identified 10 species communities and 16 functional modules, and within the communities, we can see the species often share the similar taxonomy or functional roles, such as short-chain fatty acid production, and pathways with the same modules were also functionally related, supporting the idea of coherent ecological units existed in the gut microbiome. Then we examined association with metal exposures. On the left, the heat map shows association between 19 stool metal elements and species communities, where color indicates effect size and stars denote FDR significance. Multiple metals were associated with lower level of beneficial communities, several, particularly communities 3, 4, and 6, which are involved in butyrate production and immune support. And on the right, the first plot shows association with functional modules, and we can see multiple metals, except sodium, were negatively associated with multiple function pathways or modules that are involved in amino acid biosynthesis and energy metabolism, suggesting the disruption of the beneficial gut microbiome functions by metals. Finally, we have assessed the links to neurodevelopments, and we can see here the higher level of a module that related to amino acid biosynthesis, which is module 14, were associated with lower ADHD-related symptoms, while module 16, a module related to fermentation functions in gut, was associated with higher symptom scores of ADHD. In the mediation analysis, sodium shows a protective association with hyperactivity that was partially mediated by a microbiome function. 14. And in contrast, metals such as barium, vanadium, and copper were associated with higher ADHD scores, with both Clostridium-dominated community and fermentation-related functions contributing to these effects. So overall, our findings suggest that gut microbiome organized as communities and functional modules rather than a single taxa may serve as a key pathway linking metal exposure to neurodevelopment in children. This raises the possibility that targeting these ecological units could offer a new strategy for intervention or prevention for metal exposure and also for neurodevelopmental health in children. Thank you.